for reproduction, in reproduction. For it to move in the female reproductive system, that cell is going to have some features or modifications which will enable it to swim and move in the female reproductive system. We give examples of specialized cells both in plants and in animals. And specialized cells in animals, we say they include, we have like sperm cells, we have the red blood cell. We also talked about the nerve cell, like this nerve cell here, which we said it is used to conduct electrical impulses. And we explained what's an impulse. We said an impulse is an electric signal that is transported or transmitted in the body. And we said that how this cell is specialized is that it has axons and dendrites, which are used in conduction of impulses. Another example of a specialized cell in animals is a red blood cell, which we said a red blood cell is used to transport oxygen and carbon dioxide in the body, and therefore the adaptation or the specialization or the modification, in other words, is to create a lot of space inside the red blood cell so that we can have as much oxygen and carbon dioxide being transported as possible. Like, for example, a red blood cell will not have a nucleus at all, so that it can have more room for packaging and transportation of oxygen. It is also biconcave, it is thin at the middle, like you can see here. It is thin at the middle compared to the edges, so that a lot of oxygen and carbon dioxide can actually dissolve in the red blood cell, allowing better transportation of oxygen. We also talked about the white blood cells, which we said their function is to protect the body against infections. And we said one of the adaptations or modification is that it has large nucleus, which is used to engulf or to break down the disease-causing microorganisms. So we have that. And we also say they are able to change in shape so that they can move through capillaries, arteries, and even arterioles. Very good. So today we also talked about tissues or organizations of tissues, which we said that tissues are made from cells. And we said that many cells that perform a similar function, collectively, they are referred to as a tissue. Very many tissues which perform a similar function or which perform functions geared towards obtaining a specific goal, they are referred to as organs. Organs, like for example, we have vascular tissue. Now, vascular tissue is a plant tissue because phloem and xylem are used for transportation function. So a vascular tissue is used for transportation of both water and products of photosynthesis or food in plants. Because both of them perform a specific function which is transport, a similar function which is transport, therefore we refer to them as a tissue. So the phloem and the xylem form a vascular tissue. Another example of a tissue in animals, now this time we have blood tissue. Okay, blood consists of platelets, white blood cells, red blood cells. So when these ones collectively, when you refer to them collectively, you will refer to them as blood tissue. Why? Because it's made up of very many cells performing almost a similar function. That is, white blood cells will protect the body against diseases, red blood cells will transport oxygen, platelets will be used in the process of blood clotting. So all of them, because they are in the blood, they are in the same system and they are performing almost a similar function, or they are functioning together to achieve a similar function. Therefore, they are referred to as a tissue, and that's why now we have a blood tissue. So today I want us to talk about cell physiology. And yesterday we introduced cell physiology, where we said that cell physiology is the study of functions of a cell. Now that we know cells are the simplest form of a living organism, like the basic unit. The simplest basic unit of a living organism is a cell. So how do these cells function? What are the roles of these functions? How do these cells relate with other cells? How do they behave when they are placed in solutions of different concentrations? That is what cell physiology is about. And because we say that each cell, be it an animal cell or a plant cell, has a cell membrane, we went ahead and studied the cell membrane structure. And we said that the cell membrane has two main functions. Number one, it controls and regulates movement of materials in and out of the cell. How is this possible? This is possible because cell membranes have poles. Like you can see here, they have poles, and these poles are selective, meaning they only allow materials which are small enough to fit to move in this in the cell and out of the cell, while if a material is bigger than the pole, it cannot fit through. That's why we're saying it controls, it regulates movement of materials because it doesn't allow everything to pass through. Only those materials whose molecules are small enough to fit in the poles. The other function of a cell membrane is that it encloses the contents of the cell. So it is like the barrier between the inside of the cell and the environment. So whatever is in the environment remains in the environment. Whatever that is inside the cell remains inside the cell, unless it is small enough to pass through the poles. So also we discussed the structure of a cell membrane. Structure basically means how is it made? What makes up a cell membrane? And you can see from this diagram here, a cell membrane is made of protein layers. Okay, we have two. We have the protein layer on the upper side and the lower side. And then we have two layers of the phospholipids. Phospholipids means there's phosphorus and there's lipids in it. So basically these are fats. So the structure of a cell membrane is made up of protein layer and two phospholipid layers. Protein layer and two phospholipid layers. So like we're going to see in the questions that we were doing yesterday before we ended the lesson, we'll see a question that is asking you what is the structure? I'm explain or discuss the structure of a cell membrane. You just say cell membrane is made up of two protein layer and two phospholipid layers. Okay, yes, and then of course we have poles which selectively allow materials in and out. Properties of a cell membrane. We said properties are basically characteristics. Okay, characteristics. So ukiona swali kwa biology na kuuliza you state properties of a particular structure, it's basically asking you to state the characteristics. And one of the characteristics we talked about was semi-permeability or selective permeability. We said permeability is the ability to allow materials to pass through. That is permeability. But then when you talk about semi, it means some materials will pass through and others won't. Why? Because of the size of the poles of a cell membrane. So therefore, a cell membrane is semi-permeable or it has selective permeability, meaning it allows materials which can fit into the poles to pass through, while those that are bigger than the poles will only remain on the inside or outside of the cell. It will not pass through the poles. Another characteristic or property or feature of a cell membrane is that it is sensitive to changes in temperature and pH. So now sensitivity means it can be affected by. When you say that something is sensitive to something, it means it can be affected by either positively or negatively. But in this case, the cell membrane has a very negative sensitivity when it comes to temperatures and pH. How so? Because it is made up of protein layers and a lipid layer, what we're calling the phospholipid layer, if in case a cell comes into contact with extreme pH. By extreme pH, we mean something like strong acids. We discussed the pH scale yesterday, and we said acids are from 1 to 6, and then 7 is a neutral, that is in a pH scale, and then 8 to 14 is a base. So if you expose a cell into extremely strong acids, then it is going to be affected by it in such a manner that the protein layer on that cell membrane will be denatured. Denatured means it will be destroyed. If you expose cells into very high temperatures, the protein layer and the lipid layer in the cell membrane will denature or will destroy that cell membrane. So we also went ahead and we talked about the other characteristic or feature of a cell membrane, which is a cell membrane has both positive and negative charges. So therefore the feature of the characteristic here is possession of electric charges, or we can also say it is polarized. When do we say that something or anything is polarized? We say it is polarized if it has both positive and negative charges. Now, negative and positive will cancel out, causing neutral, right? So therefore, a cell membrane is polarized. It has positive and negative charges. Why is this important for a cell? When the cell membrane has negative and positive charges, it is very important because it controls or allows movement of materials. So it affects how materials will move in and out of the cell. So the importance of polarity is that it controls how materials move inside and out, and also the cell is able to detect changes in the environment. If it is the temperatures that are changing, you'll be able to feel. If it is pH that is changing, you'll be able to feel, because the cell will start getting irritated and having irritation will start coming about. That's why if you move closer to a source of heat, you'll start feeling the uncomfortableness of that heat. Is because your cells are made of negative and positive charges, or they are what we're saying polarized. Okay, now we went ahead and talked about difference between a cell wall and a cell membrane, and we said that a cell membrane is semi-permeable while a cell wall is permeable. What does that mean? It means cell membrane will control what passes through. If the material is bigger than the pole, it will not pass through. But for the cell wall, it allows anything and everything to pass through. So anything that can pass through the cell wall will pass through. But when it moves to the inside where we have the cell membrane, it might not pass through because a cell membrane is semi-permeable. So therefore, a cell wall will allow all sorts of water, sugar molecules, salt molecules, and even water to pass through. But for the cell membrane, only a few materials of those will pass through. If the sugar molecules are bigger than the pole, they will not pass through. But through the cell wall, they will. So then we need to talk about adaptations of the cell membrane to its function. So adaptations of the cell membrane to its function. So it has poles for semi-permeability, that one we said. So it is thin to reduce distances, hence faster movement of materials. So it means cell membranes are not thick. They are very thin for diffusion. We will talk about diffusion later in a few minutes probably, or even during the next lesson. And then it has electric charges, that is, it is negatively and positively charged, so that it can detect changes in the environment. Now, yesterday before we ended the lesson, we were doing these questions here, and I want us to begin with the questions before we introduce something else. Now, name the components of a cell membrane. That was the first question we did. So in short, you've been asked, what are the structures? What makes up? What is it that is found in the structure of a cell membrane? Name the components of a cell membrane. What makes up a cell membrane? And we said there are two things that make up a cell membrane. That is the protein layer and the phospholipid layer. Question number two. Give two functions of the cell membrane. What are the functions of a cell membrane? There are still two, and we've talked about them, and we have rewinded them today in the morning. And we've said these two functions are I don't know if there's someone who is comfortable answering that question. I want to give someone an opportunity to answer the question. Give two functions of the cell membrane. Anyone? Functions of the cell membrane. Anyone? Okay. So if there is no one, then I think I will just proceed. Now we've said the two functions of a cell membrane. Sorry?
Can I try?
Yes, you can. Yes, you can, of course.
The first one is to control the movement of materials in and out of the cell, and the other one, it encloses the cell contents.
Very good. Those are the perfect answers for that question. James, I've seen you wanted to answer. You'll answer maybe the next question, yeah?
Okay, mwalimu, asante.
Sawa, asante. How we proceed? Now, it is true. We've been told that the functions of the cell membrane ni number one, to control movement of materials in and out of the cell because of the poles. And number two, encloses. It's like our skin. You see, like the way our skin encloses the internal organs is the same thing the cell membrane functions. So number three, then that question is, explain how the cell membrane is adapted to its function. Explain how the cell membrane is adapted to its function. Now, in short, this question is asking you to explain the features of a cell membrane. The features, because an adaptation is possession of a certain feature which will enhance the way a cell membrane or an organism functions. Like, for example, let me explain kidogo. For example, if you live in the deserts, or if you live in very cold places, like in, let's say, European countries, you realize your body will become adapted to that environment in such a manner that you'll start growing long hairs on your body to enhance the heat retention, like that. So when a question asks you to explain how anything is adapted to its function, in short, unaulizwa what features are in that organism and how do they assist that organism in surviving. So hapa swali ni explain how the cell membrane is adapted to its function. So what are the features of a cell membrane and what are the functions of those features? James, do you want to go first?
I can say they are selectively permeable. Yeah.
Yeah, very good. That's a very good answer. A cell membrane is adapted to its feature. How? By being selectively permeable. It allows materials to pass through that are able to pass through, and others which are large will not pass through. Now, question inauliza to its function. So you see that answer we've been given is connected to the function of a cell membrane, which is controlling movement of materials. So if it is not selectively permeable, it would not be able to control movement of materials. But now we're saying it is semi-permeable. That's an adaptation to which function of selective permeability. Anyone else?
Another one is
Go ahead.
It is flexible and thin.
Very good. It is flexible and very thin.
To change the shape, allow the movement of substances.
Yeah, very good. Very good. Very good. Now, the fact that a cell membrane is very thin, it is to allow movement of materials easily. Like, for example, if it is thick, it would take a longer distance. Let me illustrate. Let me try and illustrate here so that you'll understand the point. So assuming that is a cell membrane A and we have cell membrane B, and materials want to move from this side to that side, and others will move in this B from that side to that side. Now if you look at the distance from this point and this point here, let me use a different illustration. Distance from that point to that point is very short compared to distance from this point to this point. So if these two were cell membranes, A and B, materials from this point to that point would move very easily and very quickly. Why will that happen? It's because the distance is short. In the other scenario, if materials wanted to move from this point to this point, it would take a longer time because of the distance. So when we say that a cell membrane is thin, we mean it has very short distance from the inside and the outside. If we say something is thick, we mean there's a longer distance from the outside and the inside. So therefore, this cell membrane that we are talking about is adapted or has a feature of being thin so that materials can move easily from one side to another, either from the inside to the outside or from the outside to the inside. So the answer here will be it is thin to increase the rate of movement of materials or to reduce the distance through which materials move. And this concept is very important because we are going to talk about it when we'll be talking about diffusion and osmosis, and especially diffusion, because diffusion will say what it is, the movement of materials, blah blah blah, and then we are going to see the importance of a cell being thin. So I hope that one is well understood and we can do the other question. Or there's someone else wanted to add something or a point, James?
Pardon?
Do you want to add another point or you good?
I'm okay.
Very good. Thank you for participating. Others also, kindly, I encourage us to continue participating. Good. The last question, explain the importance or significance of the electric charges or the polarity of the cell membrane. Now this question has been, they have slashed this question to show you different ways in which someone's microphone is on. James, yeah. Kindly mute, James. Thank you. Hi, so we're saying question number 4, it is showing you different ways in which this question can be tested. So in some cases, you'll get a question asking you, explain the importance of the polarity of a cell membrane. Or another question can be posed as, explain the significance of the electric charges in a cell membrane. So you can get a question asking you to explain the significance or the importance. So importance and significance is the same. So explain the importance of polarity of a cell membrane. Tulisema polarity is the possession of the positive and the negative charges. So is there anyone who's ready to tell us what is the importance of a cell membrane having positive and negative charges? Why is it important for a cell membrane to have positive and negative charges? Anyone?
It regulates. Regulate the entry and the exit of substances.
It is able to regulate entry and exit of substances. What else?
It also protects the cell contents. Yani ile contents zote zenye ziko kwa hiyo cell membrane inaiprotect.
Inaiprotect, eh?
Yeah.
But you see, James, hii swali narrow down to polarity.
Polarity.
Polarity, like the possession of electric charges. Unaona hii polarity hii? Like significance of electric charges, eh? So in short, the question wants you to be very specific on importance of these polarities. So the first point which you said that it is able to regulate movement of materials in and out of the cell, that is correct. That is the first point there. The second point, anyone else? Let's see if there's someone else who will get it. Hold on, James, and then I'll give you the second point, isn't it?
Yes.
And the other learners. Anyone else who is willing to contribute? We've said the significance of electric charges is number 1, to regulate movement of materials in and out of the cell. Anyone else? Good. If there isn't, let me give the second reason. You see, in biology, when you see 2 marks in a question, that means you're either supposed to give 2 points or 1 with an explanation, with a very good explanation. So it's good you note that. So apart from regulating movement of materials outside the cell because of the possession of electric charges, the cell is able to detect changes in the environment. And that is the most appropriate answer here. So explain the importance or significance of electric charges in a cell membrane. So you'll say the cell is able to detect changes in the environment. The cell is able to detect changes in the environment. So when the temperature changes, the cell can be able to detect because it has negative and positive charges. When the pressure changes, the cell is able to detect. So the explanation here will be the cell is able to detect changes in the environment. The cell is able to detect changes in the environment. So thank you very much for your contribution. I greatly value that contribution, and I'm happy to hear that we are learning, and that is very encouraging. Now, on to something that is very, very important in biology. Actually, as a matter of fact, this is where biology begins to biology. The concepts of biology begin to actualize here. So here we are going to talk about processes which are which are common in cells in relation to materials that move in and out of the cell. Also some molecules, the way they move from one place to another. Very interesting concepts. So physiological processes. Physiological processes. So the movement of materials across cell membrane is facilitated by physiological processes. So physiological processes are basically ways in which materials will move in and out of a cell membrane. Okay, so materials can move in and out of the cell membrane through number 1, diffusion, number 2, osmosis, and number 3, active transport. Ways through which materials move in and out of a cell are what we call physiological processes. So number 1, the first physiological process is diffusion. Number 2, we have osmosis. And number 3, we have active transport. So what we are going to be doing for the next few lessons is we'll be looking at each and every physiological process. We discuss it, and then we will even do questions on that physiological process. So tukimalizana na diffusion, we will have one lesson specifically meant for questions which can be set in diffusion. Once we are comfortable with it, we'll move to osmosis. Osmosis is quite deep because it has a lot of things to cover, but we will do it, and we'll do very many questions on it. Once we are comfortable, we move to active transport. And so we begin with diffusion. So the first thing we need to do about diffusion is to define or state what it is. So diffusion is the process by which particles or molecules move from a region of high concentration to a region of low concentration. Diffusion is the process by which particles or molecules move from a region of high concentration to a region of low concentration. I will use a very good example, and the example that I'm going to use is an example of perfume or cologne. Now, I'm sure you've been in a situation where someone has boarded maybe a matatu that you are in, and after a few minutes, the whole of the matatu is full of the perfume that they have worn. Why does that happen? You see, the perfume that you wear or the cologne that you wear, those molecules, because you spray them on your body or on the clothes that you're wearing, those molecules are very highly concentrated. The molecules of that cologne, they are highly concentrated on your body or on that specific part where you sprayed them. So what happens across during the day, or what we call that you've been freshened through the whole day, it's the molecules which are highly concentrated from that place where you sprayed your cologne. They'll be the one that will be moving from that region of high concentration, and they'll be getting spread across the environment, causing that smell or that aroma to be felt by everyone who is around you. Okay, so therefore, when you spray, for example, cologne on your trouser or on your shirt or your skirt, that cologne has very highly concentrated molecules of that particular perfume. And because they are highly concentrated in that area where you have sprayed it, the molecules will start spreading. Pole pole too, they will spread. So when you move in a room, because where you are, there's high concentration of that cologne, it will start spreading to areas where we don't have that cologne. So eventually, at the end of the day, everyone in that room will be able to tell there's someone who's wearing this specific type of cologne. Another example, sometimes you can walk through the day and it is very hot, you have carried out very vigorous exercises, umepitia mahali kuna vumbi, umekuwa ukitembea, umesweat, a lot of things and then ufike kwa nyumba utoe kiatu. Ukitoa kiatu uweke karibu na shoe rack. A few minutes later, room yote unasikia hiyo kiatu kuna harufu kidogo inatoa, harufu fulani ya kiatu inatoa. Hiyo harufu ilitoka aje kwa kiatu ikaspread kwa nyumba? Ni kwa sababu inside that shoe there is that smell and it is highly concentrated in that shoe. So therefore what happens is when molecules are highly concentrated in a particular region or a particular space or place, they tend to start spreading and moving to where they are not highly concentrated. So from the shoes, those are that smell, not those, that smell will start spreading across the room. So eventually, after a few minutes, everyone in the room will tell that person has had a very long day. So that is what or how diffusion takes place. Molecules or particles will move from where they are highly concentrated, mahali ni mingi, and they will move to a region where there is low concentration of those molecules. So I don't know if I can illustrate using a small diagram. So we can have very many molecules of a particular, let's say a dye or a cologne. So they are highly concentrated here. So we have two regions. We have region A and we have region B. Now in region A, as you can see, the red dots that I'm drawing here, in this region, we have very many molecules of a particular cologne or anything. Okay, so between these two regions, you will see in region A, we do have a lot of particles or molecules of, let's say this is a cologne. While in region B here, we don't even have a single molecule of that perfume. So therefore, naturally, the cologne molecules or the perfume molecules from point A will start moving to point B where there is a lot of space. So the molecules will start moving to part B, and therefore part B will start experiencing that smell, or you'll start, if you're in region B or part B, you'll start smelling the cologne of the person who is seated right on in part A. Let me use a layman's example. If, for example, you're living in a place where maybe, for example, it's the rentals or maybe at home and the population increases immensely. So kitambo mlikuwa mnaishi watu wanne kwa nyumbani kwenu ama kwako, alafu upate mmekuwa wengi, now you are like 10. All of a sudden there's no space. All of a sudden you have to queue maybe to go to the washroom. All of a sudden you have to queue to go and warm water, or maybe, for example, to go to the fridge and fetch something to eat. Now the population becomes so much hectic such that some people will opt to move out of the house. Why? Because all of a sudden resources are not easily available. All of a sudden the amenities are not enough for everyone. So therefore people will tend to move from that place where we have a lot of people and go live in a place where we don't have more people, such that they will be able to have maybe their own space, they'll be able to access the amenities, and they'll be able to do things freely. So therefore, it's a similar thing that is happening here. When materials are highly concentrated in one region or in one place, or molecules or particles for that matter, they will start moving to a place where there is low concentration, and that is the concept of diffusion. Movement of particles or molecules from a region of high concentration, kwenye kuko na mingi, to a region of low concentration. I hope that concept has been understood because it is very important. So movement of molecules or particles from a region of high concentration to a region of low concentration. There's another concept that I want to introduce to you, the concept of concentration stroke diffusion gradient. Concentration or diffusion gradient. Concentration or diffusion gradient. Now, what is concentration gradient or what is diffusion gradient? Diffusion gradients come from the word diffusion, which we've said it is the movement of molecules from a region of high concentration to a region of low concentration. Now, if you were to compare region A and region B, these two regions have a very huge difference in concentration, in such a manner that in region A, we have very many molecules of, for example, let's use cologne. In this case, we're using a cologne. So we have very many molecules of cologne in region A, while in region B, the molecules of cologne are very few. So therefore, what is diffusion gradient? Diffusion gradient is basically the difference in concentration between two regions. Like in region A, let's assume we have 100 molecules of the cologne. In region B, we have 20 molecules. That's an assumption. So from the two regions, you can see region A is highly concentrated with the molecules of cologne, ziko. In region B, the molecules are 20. So therefore, what is the difference between these two regions? The difference is 100 minus 20, which is 80. So in this case, what is the diffusion gradient? 80. So the difference between region A and region B is 80 molecules. So therefore, diffusion gradient is the difference in concentration between two regions. So in one region we have high concentration, in another region we have low concentration. So that difference is what we call the diffusion gradient. Now it is very important to understand the concept of diffusion gradient, because so long as there's a difference in concentration between two regions, molecules will always move from a region of high concentration to a region of low concentration. For example, let me explain that point. I'm repeating the point I've said. So long as there's a difference in concentration between two regions, diffusion will always take place, because that means there's one region that is highly concentrated and there's another that is lowly concentrated. Let me illustrate. So we have region A and we have region B. The only way that materials will not move from one region to another is if the concentration is the same. Like for example, if we have 4 molecules of a cologne here and 4 molecules of a cologne on the other side, it means the concentration in region A and the concentration in region B is equal. So therefore, hakuna particle na molecules mingi kushinda ingine. So at this point, or in this case, diffusion will not take place. Why? Because diffusion takes place when molecules move from a region of high concentration to a region of low concentration. Now in our case here, we have 4 molecules of the cologne. In region B, we have 4 molecules of the cologne. So therefore, what does that mean? That means that the number of molecules in A and B are the same. So we cannot have a region that is highly concentrated than the other. Both are equally concentrated. So in that case, there will be no diffusion. But in a situation where, let's assume in region A, we have 15 molecules of the cologne. We have 15 molecules of the cologne. In region B, we have 5 molecules of the cologne. the difference in concentration between region A and region B is 10. So therefore, automatically, which is the highest, the region with high concentration? The region with high concentration becomes region A. The region with low concentration becomes region B. So therefore, we will have molecules moving from region A to region B because region A is highly concentrated than region B. So the higher the concentration gradient, or the higher the difference in concentration between two regions, it means we are going to have a very high rate of diffusion. So diffusion itakuwa kubwa, rate yake itakuwa kubwa mahali ambapo kuna very high concentration gradient. Like for example, if the difference in concentration is 1000 molecules, tuseme region A iko na 2000 molecules, region B iko na 2 molecules, difference itakuwa 1999. So because that gradient is so huge, the rate of diffusion will be very high. If the difference is maybe 2 molecules or 3 molecules, the rate at which the molecules will move will be very slow. So therefore, the rate of diffusion can be affected by the concentration or diffusion gradient in such a manner that if the diffusion gradient is high, the rate of diffusion is very high. If the concentration gradient is low, then the rate of diffusion is low. Now, I hope you have understood that concept of diffusion and diffusion gradient. The reason why I spent most of the time there is because if you do not get to understand what is diffusion, then you're going to have a challenge with the whole subtopic. So I want, by a show of maybe raising hands or maybe typing understood, kindly type for me if you have understood diffusion, what it is, and the concept of diffusion gradient, or if you haven't, kindly type for me, because I do not want to move on from this if people have not understood, because it will make no sense if I move on to talk about diffusion and we have not gotten the definitions and the concept of concentration gradient. So kindly give me feedback. Yes, I've seen Hope has understood, very good. Maura has understood. Wengine tumeelewa? I want to believe we have understood. Very good, very good. So therefore, let's move on to the next concept, still under diffusion. The next concept here is a practical activity. And the practical activity is a practical activity to demonstrate using potassium permanganate 7 to demonstrate diffusion. But since we are not in a position to do that practical activity, we can hold that practical activity together with the one we talked about last week. No, the last lesson that we had, apart from yesterday, the lesson we had in the morning. We say that we are going to hold a practical on observing the cells of an onion using using a microscope, or in short, preparation of a temporary slide. So we said we are going to do that, so we can combine that with this activity so that we'll be able to see how diffusion takes place. But in short, I don't know if you guys know potassium permanganate 7. Potassium permanganate 7 is purple in color. Inakuanga purple in color. So if you take potassium permanganate 7 and put it in water, the color of the water changes to purple. So you're going to require potassium permanganate 7 crystals. You will require glass tubing. You'll require 100 centimeter cubic beaker, and of course water. Procedure: hold a glass like this one. You hold a glass tube with a thumb vertically, then you add potassium permanganate 7 in that glass tube, alafu uweke maji like in step 1. Let me show. I think it's better if I illustrated. So we have this one here, the first one. So we have a glass tube. This is the glass tube. We put or place potassium permanganate 7 inside the beaker without the water. Then you should pour in water. When you pour water in, we move to step 2, where you remove the glass tubing. As you remove the glass tubing, hapa chini utaona tunabaki na potassium permanganate 7. And remember, potassium permanganate 7 is purple in color. So what happens is, immediately you remove the glass tubing, the particles of potassium permanganate 7 will start spreading in the water. So at the beginning, the potassium permanganate 7 molecules will be highly concentrated pale chini kwa glass, kwa bika penye ulikuwa umeiweka. But with time, the molecules will start spreading now across the whole beaker. So after a few minutes, you'll realize the whole of the beaker has turned purple. Why has the water turned purple? Because of the molecules of potassium permanganate 7 spreading inside the beaker. So you take potassium permanganate 7, which is purple, unaiweka at the bottom of the beaker, and then ukiongeza maji, the molecules will start spreading all over through the process known as diffusion. So one way in which you can demonstrate diffusion in a laboratory is using potassium permanganate 7, and the reason why we use potassium permanganate 7 ni because of the purple color. So when that purple color spreads, then we say diffusion has taken place, as the molecules have moved from the bottom of the beaker where they are highly concentrated and they spread across the beaker. Okay, that's one way in which you can demonstrate diffusion. Another way in which you can demonstrate diffusion, and this one I want you to try at home. Kindly try this one at home. I want you to use a cologne if you have a cologne. So today, or after you leave from work, you come back home, nataka uende sitting room. In one corner of the sitting room, you spray some perfume or cologne. Uspray hapo kando, hapo kona, alafu you settle down and continue doing whichever chores you're doing in the house. So unaenda kwa kona moja ya nyumba, una spray cologne yako kwa kona, alafu you leave and you continue doing your chores. One thing you're going to notice is when you spray in one corner, as you do your duties, the room will start filling up with the cologne smell. Okay, why is that happening despite the fact that ulispray pale kwa kona? Those molecules, because they are very highly concentrated in that place where you sprayed, they will start spreading in the room. Why are they spreading? Because of the process known as diffusion. For those of us that for those people that are lucky enough to have cars, or those people that have been maybe transported from one place to another by an Uber, or they have boarded a friend's car, nowadays I've seen a trend where people have some perfumes spread, some perfumes hanged pale kwa I don't know, the rear mirror of the car. Kuna ile kioo fulani pale kwa dereva kwa windscreen, unapata most of the people nowadays wamehang ka cologne, kachupa kakona perfume kamehangiwa hapo. So inaweza kuwa na lemon, inaweza kuwa na strawberry, inaweza kuwa na whichever is their favorite flavor, so wanahang hapo kwa hiyo kioo yenye iko hapo ndani kwa windscreen. And then ukiingia kwa gari, unasikia hiyo gari it has a very interesting smell of, if it is lemon, if it is strawberry, it will be smelling like strawberry all over. So the same concept is being borrowed. So hako kachupa ama hako ka sachet kakona hiyo flavor, alafu because of the heat and the temperatures being high in that vehicle, that cologne or that perfume will start spreading in the car. So ukiingia tu kwa gari hivyo utasikia ina smell hiyo lemon ama ina nukia hiyo strawberry. So it's the same concept that is being applied. So that's another way of testing diffusion. So you can test diffusion using those two ways. You can use potassium permanganate 7, or you can also use cologne, and you'll be able to feel or smell the smell from that place where you placed it with high concentration. It will spread to other regions of the house where you did not spray your cologne. So that is basically how diffusion takes place. Good. So diffusion, we can therefore conclude that in diffusion, molecules move from a region of high concentration to a region of low concentration. There's another practical activity that is very common, and this practical activity is highly examinable. Most of the times they tell you to do it in paper 3, and if they don't tell you to do it in paper 3, they have found a way of testing questions from this area and testing it in form of a question in paper 1 ama paper 2. Now this practical activity is demonstrating diffusion using a whisking tubing. Now what is a whisking tubing? A whisking tubing is a synthetically made piece of paper which performs similar functions as a cell membrane. You see, the good thing with technology is that when we realized that cell membranes are selectively permeable, some scientists invented a piece of paper, it's nylon, and that piece of paper functions just like a cell membrane. The only difference is a cell membrane is natural and a whisking tubing is synthetic. So like in this diagram that I'm demonstrating here, you can see the whisking tubing. So this is the whisking tubing, this one here, hii ndio inaitwa whisking tubing. So basically ni karatasi imetengenezwa kwa lab ambayo you can put materials inside and tie on one side, alafu you tie the other side. So whatever is inside can easily move in and out because the whisking tubing is semi-permeable. It functions just like a cell membrane would. So we can demonstrate diffusion using a whisking tubing. So what will we require? We will require whisking tubing, which is 8 centimeter long. We will also require 5 ml starch solution. 5 ml starch solution. Now note starch solution. Now you will realize later that the reason why we are using starch is because starch has very large molecules. Molecules kubwa haziwezi zikapita through the poles of the whisking tubing ama cell membrane for that matter. Okay, then we will require this dilute iodine solution. Now last time we had a lesson, we talked about iodine where we said if you've ever been injured na ukapelekwa hospitali, kuna ile dawa fulani ya brown unaoshwa nayo, that is iodine. So iodine is usually brown in color and mostly it is used in a medical field to clean wounds or to disinfect surfaces. So we will be required to have iodine. We'll also be required to have a glass rod and a dropper and water inside a beaker. What is the procedure? Tie one end of the whisking tubing using a thread. So unachukua whisking tubing, unafunga side moja, okay? You tie it on one side using a thread or a string, the way they have done here. So ukiangalia hapa utaona kuna hiyo part, it has been tied. So when you tie one end, what is the next step in the procedure? You are supposed to pour starch solution into the whisking tubing. So ukishafungua hiyo karatasi tunaita whisking tubing, ufunge side moja ya chini. Through the other side which you have not tied, you pour starch solution inside the whisking tubing. After you pour, what do you do? Sasa hiyo part yenye umetumia ama side yenye umetumia kupoa starch ndani, you also tie it. So at the end of the day, you're going to have something like this. Something like a whisking tubing that has been tied on both edges, okay? So it has been tied on this side and on that side. Inside the whisking tubing, you're supposed to have starch solution. Why? Because you poured starch solution inside the whisking tubing. So procedure tumesema, you take a whisking tubing, you tie one side, and through the other side you pour in starch solution on the inside. Alafu kwa bika huku unaeka iodine solution. You see, like in the illustration here, kuna iodine solution which is brown in color. So you pour iodine solution in the beaker, not inside the whisking tubing, but in the beaker. Immediately you do that, there are two things you're going to notice. You will notice that the color of iodine is brown and the color of starch solution is white. You must notice that because starch solution ni kama tuseme uchukue unga and then ama glucose, no, not glucose, unga, and you dissolve it in water. Okay, so it will have a whitish. Unaona hata ukipika ukipika ugali, si unaona maji na turning almost milky because of the starch in the maize flour. So when you pour starch solution inside the whisking tubing, you will note that the starch solution's color is white while iodine solution is brown. Why am I saying you have to note that? You have to note that because at the end of the experiment, there will be a color change. Okay, so at the beginning of the experiment, the iodine in the beaker, will be brown in color. Starch solution will be white. So when you give it time, what do you expect will happen? Because we are talking about diffusion, two things are expected to happen. For example, you expect either starch solution will leave the whisking tubing and move into the beaker. That is the first expectation. Or if that does not happen, then iodine solution will be the one to move inside the whisking tubing. So it is either starch solution itoke kwa whisking tubing iende nje kwa beaker, or it is the iodine which will move from the beaker into the whisking tubing. What will determine what will move? It will be iodine molecules that will move into the beaker if they can fit through the small pores of the whisking tubing. If the starch molecules ndizo ndogo kushinda the whisking tubing poles, they will be the molecules of the starch to move from the whisking tubing to the outside of the whisking tubing. So what dictates what is going to move? It is the size of the molecules. Now let me tell you what moves. It is the iodine that moves from the beaker into the whisking tubing because the molecules of iodine are very small, smaller than the whisking tubing poles. But for starch solution, the molecules of starch are so big haziwezi zikapitia through the whisking tubing. So therefore, at the end of the experiment, you expect that the molecules of starch solution will move from the outside of the whisking tubing, to the beaker. Okay, we are expecting starch molecules will leave the whisking tubing and we are expecting iodine molecules. Let me correct that. That was wrong. Let me correct. I've said comparing the iodine molecules and starch molecules. I'm repeating kindly. Comparing the iodine molecules and the starch molecules, iodine molecules are smaller. They are smaller compared to the starch molecules. So therefore, starch molecules will be retained inside the whisking tubing, while iodine molecules will move through the poles of the whisking tubing and they will move inside the whisking tubing. Remember nimesema a whisking tubing functions just like a cell membrane by selectively allowing materials to move in and out. But because the starch molecules are larger than the poles, it is the iodine molecules will move in. So give me a minute kindly nimalize because we are almost done. Now what will be the effect of the iodine molecules moving into the starch solution? The color will change. Remember I told you it's good you note the color of iodine and the color of starch solution at the beginning. So when starch molecules come into contact with the brown color of iodine, they trigger a color change from brown to blue-black. Okay, I've said when starch molecules come into contact with iodine, they trigger a color change from brown to blue-black. So therefore, at the end of the experiment, you expect that in that beaker, the color of iodine will remain the same, brown. But the color of the solution inside the whisking tubing will have changed. It will have changed from white to something we call blue-black. Okay, and when you're writing blue-black, you should always include a hyphen. Okay, this blue black, that one, you should always include a hyphen because it is two colors in one. So like when you spread it, you'll see the shade of black and the shade of blue. So we say it is blue black. So what observation will you make? The contents of the whisking tubing turned blue black because the molecules of iodine were small enough to pass through the whisking tubing compared to the molecules of starch, which are bigger than the poles. So the observation, the contents of the whisking tubing turned blue black and the contents in the beaker remained brown. Why? The wall of the whisking tubing is semi-permeable, hence allowed small iodine molecules to pass through it from the beaker, okay, while starch molecules remained inside the whisking tubing. So therefore it meant contact of the starch solution with iodine formed the blue-black color. So the explanation is, since the molecules of iodine are so small, they passed through the whisking tubing, while the molecules of starch remained inside the whisking tubing. So therefore, the color of starch solution changes from white to blue-black, or we can say the color of iodine was changed by the starch solution to blue-black. So at the end of the experiment, you expect the beaker's iodine in the beaker will retain its color, which is brown, but inside the whisking tubing, the color will change from white to blue-black, simply because iodine molecules are very small, they're able to pass through the whisking tubing. That's another way you can actually test for diffusion. Now, before we talk about factors affecting diffusion, today in the group, I'm going to send a question related to introduction to diffusion, related to diffusion gradient, related to this practical activity about testing for diffusion using a whisking tubing. Then we'll discuss that question, and once we are comfortable with the questions, then we can talk about factors affecting the rate of diffusion. What causes diffusion rate to be high or to be slower? So expect today that I'm going to, immediately after this, I'm going to send a few questions based on the introduction to diffusion, based on diffusion gradient, and the practical activity that we have done using the whisking tubing to illustrate how diffusion can take place. So if there is no question, our time is up. We will proceed from there tomorrow in the evening. But if there's a question, I'm ready to answer. Yes?
We've learned that diffusion is the process by which particles or molecules move from a region of high concentration to a low concentration.
Yes.
Now, my question is, what will happen with the diffusion when the concentration are equal on both sides?
Now, if we have two regions, like we have this region here, A and B, and the concentration is equal in both sides, there will be no diffusion. So therefore, it means, or like for example, kama ilikuwa colon, it means the room is already full of that colon. So you already feeling that smell already. You can smell it, right? So there will be no movement of materials. So if there is equal concentration, diffusion does not take place. Diffusion only takes place where we do have a difference in concentration. That's why I said the concentration gradient concept is very important, because if there is equal concentration, diffusion does not take place.
Okay.
Yeah.
Thank you.
Thank you too for the question. Yes?
Yeah. Yesterday as I was revising, come across another question. Ilikuwa inauliza, it was a practical, onion, a piece of onion. Then you have been told to calculate determine the length of each cell in micrometers. Sijui kama ulishafunza maybe.
Now what we did because we said we need to, so we said we are going to have practical sessions.
Yes.
We decided that we'll skip that concept
for practical,
so that we will do it together with the practical, because we must do that practical about observation of an onion cell. So once we're able to observe it, we'll even be able to count the cells and estimate. By then nikahesabu ka simple sana. But I figured it's only right if we do that when we do the practical, because you'll understand where the concept is coming from, because you'll be able to understand what is the field of view, how to count the number of cells, and then we will learn how to estimate the size. So ni kitu tutafanya pamoja na hiyo practical, and it is very simple, isikupe stress. Once we do the practical, we will be able to do it, and it is very simple. Ni venye nilikuwa nataka kuifunza pamoja na practical kwa sababu they work very well together.
Okay.
Yeah.
Thank you.
So yeah, James, ulikuwa umeniambia you'll organize on how we can do the practicals maybe over the holiday or midterm. I'm trusting that you have a WhatsApp group you can communicate so that we can plan on the same, because sasa unaona tuko na practicals mbili tumeprepare sasa. Tumesema tutafanya practical ya diffusion using Whisking tubing, na sasa kuna hiyo ya observation using a microscope, because kama last year waliletewa swali ya kutengeneza temporary slide, and you never know if in 1 year's time wanaweza kosa kuleta this year, next year walete. So it's only right if you're able to use a microscope and to prepare a slide.
Alafu mwalimu, kulikuwa na another one ilikuwa ina require you draw type of muscles. It is also practical. You draw a type of 3 types of muscles, then you label, and you draw, then you label. Now my question was when you're drawing, are you supposed ukiangalia kwa textbook kuna vile kuna pita hizo some lines zinaitwa sijui I don't know, the word is somehow hard. Yeah. Can you add them when you're drawing, when you're making your practical?
Okay, no problem. We will draw. One thing I know we cannot be able to see is we cannot be able to see those cells, not unless we use an electronic microscope. But I can explain further. That is Form 4 work, by the way. I'm glad to hear you reading ahead. Hiyo ni kazi ya Form 4 uko mwisho, the 3 types of muscles. So we will still look at them. So I don't know, whom do we appoint to be in charge of what we are going to do? Mwenye atatuchukulia record so that we can liaise with them to organize, tujue the time we are going to do the practicals and which practicals we are going to do. Can we have one of us as the one in charge?
See, I thought you appointed James.
Okay, James.
Yes.
Yeah, you are in charge, eh?
Okay, I think we shall form a group, students group,
okay.
And we shall discuss much on that and other things.
Sawa. Okay, in the meantime, tuandikia hapo, we will do a practical activity on preparation of slides, a practical activity on diffusion using a Whisking tubing.
Ya pili ni?
Practical activity to demonstrate diffusion using a Whisking tubing.
Yes.
And then uongeze hiyo part ya types of muscles as we've been requested by Monica, muscles, so that we can learn how to draw the muscles, the smooth, the cardiac, and the skeletal. So types of muscles.
So far there are 3.
There are 3. Yeah, there are only 3. Tunakuanga na smooth muscles, and then we have the skeletal and the cardiac. Cardiac ni za moyo, za roho, zenye zinakuanga kwa roho. Skeleton ni zenye zinakuanga zimeshikwa kwa skeleton, like hapa kwa mkono ama huku nyuma kwa mguu. And then of course we have now the cardiac muscles za roho, because za roho hazifanani na zingine. Na pia hizo zenye smooth ni kama zile ziko kwa digestive system. Unaona hapa kwa esophagus, when you're swallowing food, you're able to swallow food easily.
Yes.
Hizo ni tofauti na zenye ziko kwa skeleton, because za skeleton zinasaidia movement. Hizi za smooth muscles, they allow in movement of materials, so that's why zinakaa, we'll see they are spindle shaped. We'll look at all those structures, but basically there are 3: smooth, skeleton na cardiac. Smooth ziko like for example kwa digestive system, skeletal kwa skeleton, cardiac ziko kwa roho. Sawa?
Yes.
are very good. So we look at that even when you're doing the practicals, and we learn how to draw them and to label them still. So thank you for that. So with that, I believe we can then comfortably end the lesson, and we make a date to meet tomorrow, God willing. So you guys have a good day. Take care of yourselves, and may you be guided and protected by God. See you tomorrow, God willing.
Good day too, mwalimu.
Okay, kwaheri.